Hello, everyone. So, this talk I gave at St Thomas's Hospital in London, and it was for the launch of the RIOT Science Club there. So I founded the RIOT Science Club, and we are based at Denmark Hill in the IOPPN, and we expanded to the St Thomas's Hospital with a tremendous help from Alex and Miguel, who are based there. The purpose of the talk was obviously to introduce people to the reproducibility crisis if they weren't aware of it before, and it was also to bring over a few skeptics onto the side. The audience was mainly neuroimaging and biomedical research. So the examples I have given and the landmark studies that have kind of given credence to the idea that there is a reproducibility crisis are kind of towards that ilk. So I apologise that it's sort of focused on that, but hopefully you can walk along with me anyway. So I'll just get into the detail. So first of all, I won't dwell too long on this slide, but basically there's a heated debate about what reproducibility means, whether it's reliability, replicability, and so on and so forth. But all I'm interested in is all of these things, and I just label them reproducibility, because if you can reproduce a study's protocol or their analysis, that's great. If you can do all those and get the same results, that's really good. And all of these things are the kind of touchstones to scientific best practice and also robustness, which of course are really important for scientific advancement. So that's why I'm just interested in a broad strokes umbrella term, and I use reproducibility. Now, I won't show the video that this links to, but I do encourage you to do it. But basically, in this video, it shows you a survey that was conducted that asked 1,500 scientists the question, is there a reproducibility crisis? And it asks all kinds of supplementary questions, such as what's contributed to the reproducibility crisis? Have you tried to replicate your own work, and have you tried to replicate other people's work, and so on and so forth? And surprisingly, or maybe disconcertingly, a lot of the responses suggested that researchers can't routinely replicate other people's work or even their own work. In this figure here, you can see that about 90% said that there was a reproducibility crisis. 52% said that it's a significant crisis. So why is it that we think this? Why are scientists saying that we're in a reproducibility crisis? Well, as I said, there's a kind of a confluence of events that happened over the past 10 years. And one of the big ones was this, which was published in 2015. And it was the Open Science Collaboration, and it attempted 100 replications of psychological studies published in 2008 in high-impact, high-profile journals. Now, 97% of the studies in the original studies reported a significant result, a statistically significant result. Now, for all kinds of mathematical sort of logic and reasoning, you would expect that if you were to replicate these studies, 89 of them, as opposed to 97, would replicate successfully if all things are sort of present and there in an orderly manner. But only 35 replicated. So I think the incorrect inference is to say that a third of the psychology literature is reliable and replicable. I think it just means that better efforts can be made to make sure that the research out there is rigorous and robust and tested and confirmed. Now, of course, this caused a splash, and it caused it was quite controversial because there was issues over how do you define a successful replication? And so there was another study conducted, the Many Labs project, which attempted to replicate 13 classical and contemporary psychological studies, and it was conducted across 36 sites, and there was over 6,000 participants. Now, just to navigate you around the graph, So we can see here the blue X's, which is the original effect size, and we can see the different sites in grey and green. And these kind of Star Wars, I forgot what they're called, spaceship thingies, that's sort of where the average effect size is. So along the x-axis you have the standardized mean difference, which is just Cohen's d, or just a measure of the effect that they measured. And we have all of the different replications here. And the take-home message really is that a lot of them were successfully replicated, so they're above this zero line, but you can see that the spread of the reported effect sizes can be quite large. And also you find that a lot of the original studies underestimated the true effect size. So if you think these are more precise because they've more and more replications, you converge on what the true effect size is. But they're also in the original studies, they overestimated the true effect size, so as you can see here. Now, as I said, the audience was neuroimaging heavy, so I just picked an example from the neuroimaging literature. There are very few examples. Replication attempts in fMRI are notoriously difficult, but these did it anyway. And they applied some sort of Bayesian analysis, which I don't quite understand. But Zoltan Dianesh has actually already done a talk for the RIOT Science Club, and his talk is on our YouTube channel. But basically, Bayesian analysis gives you three pieces of information. Whether your data shows that there's evidence in support of the alternative hypothesis, which is usually good news, or the null hypothesis, so there's sort of evidence of absence. And then the third is whether your data is insensitive, so it's just not picking up the effect because it's not adequately powered, you need a bigger sample size. So they tried to replicate 17 studies that looked at brain structure behavior associations, and only one of them showed a result that was in favour of the alternative. 8 of them actually showed evidence in favour of the null, and the others were, I guess, insensitive. So those are just sort of landmark kind of cold water splashes to the face that kind of made everybody realise that, okay, there's something needs to be done. So moves were made to try and identify, well, what are the causes to the reproducibility crisis? And it was sort of two of the culprits were incentives. So research is conducted in a highly competitive field where publication and grant funding track records are enormously valued. And these can undermine confirmation, replication, and verification attempts. But there's also questionable research practices. So Dorothy Bishop did a wonderful article here, just this one page. It's very quick and easy to read. And she named and shamed 4 questionable research practices that really do not help. Publication bias, low power, p-hacking, and HARKing. So what I'm going to do in the next portion of my talk is just unpack these things, starting with the QRPs, the questionable research practices. So first of all, low power. So power is statistical power is basically what is the probability of you finding the effect that you expect given your sample size, the effect size that you expect, the alpha and beta level. So power is really important because if you're not adequately powered, you could miss an effect, or you could find an effect that wasn't actually true, or an over or underestimate of the effect that you're seeking. So it's very, very important to effectively distinguish noise from signal. So Kate Button and colleagues have what they do is they seem to make a point of trawling through the literature and finding out, how powered are the studies that we're conducting? Are they underpowered, or are they overpowered, or are they adequately powered? Now, typically we would like to think that our studies are powered at about 80%. So we'll be 80% or 80% probability of finding the effect that we want. That's what we kind of accept. And what they found in this study on the left, which is a wonderful paper, is that the median statistical power of studies in neuroimaging is 8%. That's a long way off 80. In this study of biomedical science, they found that it's anywhere in approximately half of the studies is anywhere between 0 and 20%, which is well below the 80% that is considered conventional. In this study, which is slightly more optimistic, conducted by Russell Poldrack, a wonderful paper, he said there's sort of good news. The number of participants in studies is on the rise, particularly for studies that have 100 participants or more. But at the minute, studies are only adequately powered at 80% power to detect effects of 0.75. Now, 0.75, for those who don't know, Cohen's d, or sort of estimates effect size, they work with benchmarks. And 0.2, 0.5 and 0.8 are sort of like small, medium and large effects. So a large effect, 0.8, I would say it's uncontroversial to say that that's an exceptionally large, and we don't really usually find those in neuroscience. We're sort of in the 0.2, 0.3, 0.4 range. So given that those findings are 0.8 would be very rare, and our studies are only adequately powered at that level, the chances are is that we're mass producing false positives, to want of a better, more subtle way of putting it. So more can be done. So we're underpowered in particularly neuroimaging. But these findings have been replicated in psychological studies and psychiatry studies and so on. So that's an issue. The other issue is publication bias. So publication bias can come in many different forms, but basically it can be a sort of I think the if you just ignore this. And because I've already mentioned this is the bit that we're after. Publication bias, sort of roughly defined, is the published literature is a biased selection of all studies that have been carried out. And it's usually biased in favour of statistically significant results. The null results are usually put in the file drawer and forgotten about, and resources are reallocated to somewhere where you're more likely to find a significant result. That's in terms of published studies. At the researcher or study level, you can get selective reporting, which is basically researchers only report a subset of the analysis that they carried out, either deliberately or unintentionally. They selectively report the work that they have done. Now, I think it's sort of taken as read that there is publication bias. Everybody knows this. Anybody that's tried to go through peer review knows that the system is rigged towards sort of more sexy, for want of a better word, stuff, novelty, innovative, groundbreaking stuff. And a wonderful demonstration of this was in an old paper by Michael Mahoney. And he sort of approached 75 journal reviewers, and they asked to referee manuscripts which described identical experimental procedures, but which reported positive, negative, mixed or no results. So these are the conditions. I won't go through all of them. The two that I will are these two. So these are just identical studies, but in one case the results are positive, so there's a statistically significant result, and in another scenario they're negative. So we have a very nice review that anybody would love. This is a very fine study. I have not seen the discussion section, but I don't see how it could be very far off the mark. An excellent paper. It definitely merits publishing. It goes on. Now let's look at when it was reviewed where there were negative results. There are so many problems with this paper, it is difficult to decide where to begin. While I have not seen the discussion section, I can't think of what would be there to say of this paper. I would hope that the authors avoid making wild overgeneralized. So it's a 180-degree difference. So that's what we're facing. Another example is presented by Daniel Fanelli, and I just want to draw your attention to these things. This is so what Daniel did was he looked at papers published between, I think, 97 and 2007. I could be wrong with the dates, but no, sorry, I think it was a 17-year period from the 90s to the early 2000s. Sorry, I don't remember the dates. And he looked at abstracts and studies that reported positive results. And he found that in psychiatry and psychology, this one here, it went from 70% of studies to 90% of studies reporting positive results. In neuroscience and behaviour, it sort of almost flattened out at about 90%, which is absolutely remarkable. And it's remarkable for this reason. In the previous slides, I was talking about power. So we would like to think that we're operating at 80% power to find the effects that we expect. So 80% of the time we will find a true effect. But we are also aware that the studies are only powered at about, anywhere between 0 and 20%. That's a long way off from 80. Now, if you look at it against these studies, what you find that 90% of studies are reporting statistically significant results, which is way, way above what we're powered to detect. We're only powered to detect positive results at about 20% if you want to be generous, but we're reporting positive results at 90%. I'm sorry to labour this point, but it's sort of staring us in the face that there is something else at play, and the likely thing is publication bias. So we're just only selectively reporting when we have hits rather than misses. Another demonstration of this, an alternative title for this slide, is Opening the File Drawer. This is a wonderful paper by some great people, Anne Sheil and Daniel Akins particularly. Anne has actually given a talk for the RIOT Science Club. And these basically reported the reports of positive or null results in the literature in standard reports and registered reports. So everybody knows what a standard report is. You submit your manuscript for peer review, and then it's published if peer review is successful. And so you can see here that in terms of support or no support for the first hypothesis, standard reports reported about 95% of positive results. Now, a registered report is where, before you've collected the data, you register your protocol or analysis plan, or both, to as much detail as possible, and you submit that to a participating journal, a journal that does registered reports. It goes through peer review, and if your peer review process is successful, you will get an in-principle acceptance, which is you go away, collect the data, write up your manuscript, submit your final manuscript again, and providing you have followed the protocol that you registered and you haven't made wildly exaggerated claims, we will publish it regardless of the results. So you have no incentive there to worry whether your results are positive or negative. So that's why you would be unsurprised to find that there are more null results reported. And in fact, you can see that 45 roughly % of registered reports report positive results, which is half of what we found in the standard reports. So there's a lot of nulls out there, but they just can't make their way to the surface. So that's low power and publication bias, and how these two can conflate with one another, and there's all kinds of problems. Now I want to talk about p-hacking and HARKing. I just treat these two the same because they're kind of both sides of the same coin, if you like, but they're also obviously overlap with the first two as well. But basically p-hacking is there are almost an infinite degree of freedom in how we analyse our data, and particularly with neuroimaging and how we process it as well. HARKing is, so that's p-hacking. So, sorry, I sort of jumped ahead. So because of this flexibility, you can analyse your results in a lot of different ways. And if you stumble on a significant effect during your exploration of the data, then that's great. Then you can publish that. Of course, I'm giving a sort of caricature of what actually happens, but it's not that much of a caricature. HARKing is where in science, you obviously draw up your hypothesis and you test it with the data. But what HARKing does is that when you look at the data, you realise your original hypothesis isn't looking too good. So you sort of take an inference from the data of more plausible hypothesis. And you kind of retrofit the data or sort of reformulate your hypothesis in light of the data, and then you report that you tested this new hypothesis that was drawn from the data, but you don't say that it was drawn from the data. You just say it as if you were wanting to confirm that anyway. And this is really problematic because science is the idea of testing hypotheses, but if your hypotheses are based on the data, it's the tail that's wagging the dog, and you can't be sure or confident that the finding that you have found is a false positive or not. So you can imagine a scenario where you are using your data. The data is actually a false positive. It's just noise that's just by chance you found a statistically significant result, and you are then using that to inform your hypothesis. And that's an issue. And if you think about the fact that if the studies are only powered at 20%, there's a lot of misses that could be misinterpreted as hits, and it's all retro no vomit, really. It's kind of awful. Now, and a wonderful satirical sort of demonstration of this was conducted by Simmons in 2011, and they set out to sort of show that you could become younger if you listen to a song. So, and they showed this in a rather sort of pungent good way, a very good way of doing it. So what they did is they just did a lot of different ways to analyse their data until, hey presto, a significant result was found. And they reported it, they wrote it up as an abstract, and in bold they reported what you would report if you wanted to conceal all of the p-hacking and sort of digging that you have done. And in the grey is the stuff that they sort of omitted and the extra stuff that they did behind the scenes. So using the same method as in study 1, we asked, and then was it 20 or was it 34 university undergraduates to listen only to either. So in that case, if you drop the only, it looks like they tried different conditions, and the one that worked it seems to be a more robust effect because they tried other ones. But if they say listen only to, then it moves to from a within subjects design to a between subjects design. So any difference could just be naturally occurring group differences. And there's all sorts of other things as well. But what they found was that according to their birthdays, people were nearly a year and a half younger after listening to When I'm 64 by the Beatles rather than Kalimba. And this was statistically significant. But then, of course, it goes on and on. So let's just move on. So the kind of equivalent study in a way, or it's not necessarily the equivalent, but as another demonstration of this was by CARP, and these looked at in a neuroimaging study. There are so many different component parts, and there's so many decisions that you can make along the way. It's sort of mind-boggling how infinite the combinations you can make in neuroimaging studies. So of course you would expect a proliferation of different ways of approaching your data, and this is what they found. So they got 241 articles, and their take-home messages here is that what they found when they basically did an audit. Sorry, I'm jumping around here, but they basically did an audit of the different things that fMRI or neuroimaging people did along the way, all of those different components, how they pre-process their data, how they design their experiment, how they acquire their data and displayed it, and so on. And what they found was that there was nearly as many unique analysis pipelines as studies in the sample. So everybody is doing something different. Now, I do know that this was from papers published in 2007 onwards, and this paper was published in 2012. So we have come a long way since then, and there are more standard ways of doing it, but this problem still remains. Now, there is two other things that are discussed in p-hacking and HARKing in the context of neuroimaging. The famous study is the dead salmon experiment. So, because fMRI studies, you have to test a lot of different comparisons, a lot of different nodes in the network, different voxels, different brain regions, and so on and so forth. If you keep on testing, you will just by chance find a result. So to control for that, you control for your multiple testing, your multiple comparisons. If you don't, you could find activity that's just an artifact, or just by chance. And a wonderful demonstration of this was by Bennett, where they took a dead salmon and they put it in an fMRI machine, and they found brain activity in this dead salmon. But when they corrected for multiple testing, the activity went away. And this was a huge wake-up call, and everybody kind of knows this study. Something, though, that you still see now and again, I can't point to any examples, but I definitely know that people do this, is circular analysis or double dipping. So without keep going to fMRI, let's just bring it to, say, EEG or MEG. So you have a lot of sensors, electrode channels, and you might not know, which one to analyze, or which collection to analyze. In other words, you don't have any clear direction sometimes. And what researchers tended to do was they looked at the sort of pattern of activity across the electrodes, and they looked to see those that were more active, and thought, okay, we've got these participants to do this particular task. We find that there's a lot of activity in this area, so that means that maybe this activity means this particular brain area is involved with that task. Something as basic as that. And so they say, okay, let's just focus on this area. So what they have done is they have looked at the data, then used the data to define how they then analyze the data. And this is what's called circular analysis or double dipping. And you shouldn't do this. How you analyze your data and your data or your protocol for analyzing your data and your data should be kept as separate as possible, if that makes sense. Because let's say, because you're kind of going into it blind. So let me just try and make this a bit clearer. This is a sort of a case where HARKing and double dipping cross over. So I said with HARKing, in that science is the idea that you're testing a hypothesis. So you are sort of stress testing your pet theory. But if your pet theory is informed by the data, you're not testing anything. You're just sort of conjuring up hypotheses from your data. And as I said before, your data could just be a chance result. Double dipping is very much the same, in the sense that if you don't go in with a clear idea of what areas you want to look at, what electrodes you want to look at, you can't be sure that the electrodes or areas that are active are genuinely active, or if it's just a case of non-specific brain activity in that particular area that seems to be consistent across participants. So if that's the case, then if you then choose to just focus on that area because it's active, then you could just be analysing a false positive brain activity, and so you can increase the chances of finding a false positive effect. A better description of what I'm trying to say will be given by James Kilner, who will talk about double dipping, I was supposed to say double dipping in EEG, on the 26th of March, and the video will be uploaded to the YouTube channel. So what I will just quickly round up with is, for those who aren't convinced that some people say that the questionable research practices are just a few bad apples. So I just want to sort of disabuse people of that idea. So there's been a few studies that have looked at the prevalence of QRPs, or questionable research practices. So this study looked at a few thousand researchers that are funded by the NIH, and over 33% of the respondents said they had engaged in at least one of the top 10 behaviours during the previous 3 years. Now, what are those top 10 behaviours? Well, I just want to draw your attention to these here. So number 10 is changing the design, methodology or results of a study in response to pressure from a funding source. So this is where p-hacking and HARKing really sort of come in handy. Overlooking others' uses of flawed data or questionable interpretation of data. And then, of course, circumventing certain minor aspects of human subject requirements. But I think I just want to draw your attention to these other behaviours which I don't want you to miss. But like inadequate record keeping. So how are we supposed to sort of reproduce other people's work when we ask them for the data? Their records are not all shipshape. Dropping observations or data points from analyses based on a gut feeling that they were inaccurate. Using inadequate or inappropriate research design, such as low statistical power, underpowered studies. So this is just NIH. In psychology, you get very similar things. So this is a survey of 2,000 psychologists, but the same things crop up: not reporting all measures, rounding down p-values. What else do we have here? Claiming to have predicted an unexpected finding. Fortunately, falsifying data was low, but basically the same things crop up in different areas. So what's really driving all of this? So as I said, I mentioned the perverse incentives or inappropriate incentives. So in the UK, we have the Research Excellence Framework, which is a framework to kind of point to this is what research excellence looks like. And it does serve a purpose, and I think it's important that we have that kind of metric there, but it has been appropriated and exploited for all kinds of dodgy things. And there are concerns about how REF defines, say, impact. You have to have an impact statement. What's your impact? And this is obviously regarding novelty, innovative, groundbreaking research. And you try and tell that to people in humanities where they can't really articulate how novel and innovative, given that it's sort of a revisionist approach to research. Whereas, relatively speaking, that's not to be pejorative towards humanities, but if you put it in the context of biomedical applied research, it's a target-rich environment for pointing at the applicability of a new drug or a new intervention and how it could have lots of... So in other words, it disadvantages unnecessarily certain disciplines over others. And this is what REF does, and it exacerbates things, and it also has infected hiring and promotion criteria, which you would hope would be based on metrics of meritocracy. But you can see here, and this is sort of an outdated version now, but there's a kind of spike of people hired because towards the REF. This was in 2014. Similar findings have been found for the 2021 REF. But basically, universities get desperate and they start hiring people with good research portfolios to bump up their REF. Which, anyway. Yeah, this is just another example of impact. So fortunately, the UKRI is removing the impact statement of grant applications. It was... they said it was for bureaucracy, but I think it was just for pressure, that it's just an unfair sort of hoop that we have to go through to get funding. And what impact is not really that well correlated with the integrity of research that's conducted. So that's a good progress. These studies, these next two papers, I don't know enough of them to talk about them, but the broad strokes picture is they try to simulate the kind of environment that we conduct our research, and they put things in like non-deliberate cheating or loafing, or publication bias, and what was the other things that they put in? So the analysis suggests that so in here you've got the analysis suggests that the trustworthiness of published science is in a given field is influenced by false positive rate and pressures for positive results. And this also presented the same. So I know this is a bit hand wavy, but the idea here is that they can give a real quite disturbing accurate portrayal of the real world in their simulated world, and their simulated world had lots of different perverse incentives incorporated in their simulation runs. And so it was a nice way to show that how the impact of these perverse incentives have on the quality of research. But taking us back into the real world and things that I can speak more confidently about is cash incentives. These are very, very clear-cut examples. So in China, there's all kinds of concerns about how researchers are incentivized. One way that they incentivize researchers to practice their research in a given way is to pay researchers who get a paper published in the selected journals. Nature and Science, obviously, high impact journal, high impact factor, high profile. If you get published there, you'll get paid $43,000. Now, this is huge sums of money, given that the average salary for a university professor is $8,600. That's just remarkable. Like, you've won the lottery, basically, if you get published. This is just another demonstration of the kind of money involved. So here we have billions of dollars. So for NIH biomedical research, we're looking anywhere between $25 and $30 billion in there. So there's a lot of money to be made in research. Now, I must make it patently clear, I'm not saying that researchers are just in it for the money, or they are just making a quick buck. I'm just saying that you have to do research, you need money, and you are often bent out of shape or unable to kind of practice research to the standard that you want, because you have to make capitulations to the real world that you conduct research in, and that world is governed by metrics such as ref, hiring and promotion criteria, publication bias in the system, and an incredibly intense and competitive research area where there's a lot of money there that's being gobbled up by other people. So you have lots and lots of pressure, which means that many, if not all, of the researchers out there, although they want to conduct the best research they can, they're very often barred from doing so. And this is sort of shown in the Wellcome Trust survey, which looked at research culture. And so, just to read this out loud, there was a strong sense that the system is not prioritising the right attributes when assessing research quality. In part, it was felt that it was driven by the ease with which things could be measured, for example, measuring numbers of publications and not measuring management and leadership. And this can lead to lower quality of research, superficial outputs, poor reproducibility and replicability, data picking, and so on and so forth. If you look at funding criteria, funders are sort of biased towards having researchers having fewer but larger grants, more short-term than longer-term grants, more emphasis on high-impact research at the expense of exploratory. And was a prevalent idea that funding criteria were a core driver of research misconduct, leading to deliberate embellishment and distortion of data. So this is kind of wrapping up and knitting together all of the things that I've discussed on the previous few slides. And another final demonstration, and I'll just sort of finish on this, was conducted by a team of researchers. And what they did was that they kind of dredged the literature to try and find out what it takes to be a PI, a principal investigator. And what they showed was that to be a PI, you basically already need to be a PI. In other words, you need to be on first author papers in high impact journals, which is very, very difficult to do. There were some positives. So if you stay in research, you're more likely to be a PI. If you get cited a lot, even in low impact factor journals, you're more likely. Try and be the first author wherever you can, and don't be in the middle of the author list on a paper. And they did have an algorithm for this where you could enter in your name, your gender, and the number of papers that you've published. And Kate Button from the Statistical Power fame, she entered in all of the papers she's got published, a name and gender, and she made a gender male or female, and she tried both, and she found that when she was male, she was more likely to be a PI, but when she put her gender as female, she was less likely, so she was not going to be a PI, which you would hope that research is a meritocracy, but it clearly is not a meritocracy. So what can be done? Now, usually in this talk, I go through all of the different solutions that one can do, but I'm not going to do that because that is going to be the job of Alex and Miguel. So this is the team at Denmark Hill, the RIOT Science Club there. And unfortunately, I don't have the talks from the St. Thomas's, but what they're doing is that they're streaming the talks from Feb to April. And so if anybody's watching and they're from St. Thomas's, please do check out these via satellite. Alex and Miguel will be putting on talks of their own, but that is to be confirmed. The other things that can be done is that you can go to the Open Research Conference at King's College London on June the 11th. We'll be talking all things about reproducibility, and there'll be stakeholders there from the Wellcome Trust and from the UK RIO and stuff like this. So though I'll sort of leave you on a cliffhanger and leave it there. So thank you very much for listening. Hopefully you took away something from the talk, and please do come back and try out all of our other videos. So